For over a century, scientists have been studying one of history's greatest mysteries. King Tutankhamun's tomb revealed secrets that would challenge everything we thought we knew about ancient Egypt. But in 2010, when researchers finally decoded his DNA, they discovered something that sparked a global conspiracy theory so wild, it's still spreading across the internet today. What they actually found might shock you even more. The story of King Tutankhamun begins in 1922, when British archaeologist Howard Carter discovered the boy pharaoh's nearly intact tomb in the Valley of the Kings. When Carter peered through that opening for the first time with his candle held high, casting flickering shadows on ancient walls, he was gazing upon something that had remained sealed and undisturbed for over 3,000 years. The moment is arguably one of the most significant archaeological discoveries in human history. And when asked what he could see, Carter famously replied, "Yes, wonderful things." The teenage ruler, who had died around 3,300 years ago at approximately 18 years old, left behind an extraordinary collection of treasures. There were thousands of priceless artifacts, golden chariots, ornate furniture, ritual objects, and most importantly, a remarkably well-preserved mummified body wrapped in layers of linen and adorned with a famous golden death mask that would become one of the most recognized objects from the ancient world. This discovery captured the imagination of the entire world in ways that few archaeological finds ever have. Tutankhamun became a symbol of ancient Egypt's glory and mystery, his face recognized on golden masks displayed in museums across the globe, on documentaries, in books, and in popular culture. The boy king seemed to embody something romantic about the ancient world, a young ruler who briefly held the power of an empire before his mysterious death cut his story short. Yet, despite more than a century of examination, study, and speculation by the world's finest Egyptologists and archaeologists, one crucial question remained frustratingly unanswered. Who was he really? Where did he come from? What was his family background? And perhaps most tantalizingly, why did he die so young? These questions haunted researchers because answering them could unlock an entire chapter of Egyptian history that remained partially obscured. For decades, archaeologists could only speculate about Tutankhamun's family connections and the precise cause of his mysterious death. The mummification process had preserved his body remarkably well over the millennia, but traditional examination methods had significant limitations. X-rays and CT scans could reveal physical deformities, bone structure, and evidence of disease, but they couldn't unlock the genetic secrets encoded deep within his DNA at the molecular level. Researchers had theories about his parentage based on historical records and artistic evidence, but nothing definitive. They could guess at what killed him based on his skeletal remains, but without the ability to analyze his genetic material, certainty remained elusive. That changed everything in 2008 when Dr. Zahi Hawass, the General Secretary of Egypt's Supreme Council of Antiquities, made a controversial decision that would alter the course of Egyptological research. He authorized the extraction of DNA samples from Tutankhamun's mummy and 10 other royal mummies suspected to be members of his immediate family. This decision was genuinely revolutionary because, for many years prior, Hawass himself had been deeply skeptical of genetic testing performed on ancient mummies. He believed the risk of contamination from modern DNA was too high, and that the disturbance of sacred remains was simply not justified by the potential scientific gain. Ancient DNA is fragile, degraded by time and exposure, and easily contaminated by handling from modern researchers. Extracting it without contaminating the results is incredibly challenging. But new advances in DNA technology during the 2000s changed his perspective. Scientists had developed more sophisticated techniques and more sensitive protocols. The potential benefits suddenly seemed worth the risks. 2 German specialists in ancient genetics worked alongside Egyptian scientists from the National Research Center and Cairo University's Faculty of Medicine. Together, this international team established state-of-the-art DNA sequencing laboratories. One lab was set up right in the basement of the Egyptian Museum in Cairo itself, and another was established at the Faculty of Medicine at Cairo University. These facilities allowed researchers to handle the delicate ancient samples under controlled conditions while minimizing contamination risks. The research took 2 full years to complete, with the team carefully working through samples, running tests repeatedly to verify results, and conducting statistical analysis to ensure accuracy. When the results were finally published in the Journal of the American Medical Association in February 2010, the scientific world paid close attention. This was the most comprehensive genetic study of royal mummies ever undertaken, representing a genuine breakthrough in the field of ancient DNA research and Egyptology. The team analyzed 11 royal mummies in total and conducted rigorous genetic fingerprinting, using advanced molecular techniques to establish family relationships spanning 5 generations of the royal family. The results were genuinely remarkable and often surprising, even to researchers who thought they understood Egypt's royal history. They definitively identified Tutankhamun's parents for the first time in history with genetic certainty, something that had eluded scholars for centuries. His father was the pharaoh in tomb KV55, widely believed to be Akhenaten, the revolutionary pharaoh who attempted to introduce monotheism to ancient Egypt during his unusual reign. His mother was the mummy known as the Younger Lady, found in tomb KV35, alongside other royal females. But here's where the discovery gets particularly interesting and unexpected. The DNA analysis revealed something that would have shocked ancient Egyptians living in that time period, and certainly disturbs modern sensibilities. Tutankhamun's parents were almost certainly full siblings. They shared the same mother and father, making them brother and sister, not merely cousins or distant relatives. This discovery confirmed historical suspicions and theoretical proposals that royal incest was practiced within Egyptian royal families to keep bloodlines pure, to prevent power from flowing outside the royal house, and to maintain dynasty stability. The practice was seen as a way to preserve the divine nature of the pharaohs and their connection to the gods. But the ramifications of this finding went far beyond merely confirming historical theories about royal practice. It helped explain a pattern of genetic problems that would appear in subsequent generations of the royal family. The DNA analysis also revealed a tragic truth about the royal family's health. Tutankhamun suffered from multiple genetic conditions, many of them likely inherited from the generations of interbreeding that preceded him. His left foot was severely deformed, a condition that would have caused him considerable pain and difficulty walking. Archaeological evidence from his tomb supported this finding. Researchers discovered walking sticks buried alongside his body, and contemporary artwork, particularly a painted relief showing him in the garden with his wife, Ankhesenamun, depicted him leaning heavily on a walking stick with one leg lagging behind. For nearly a century, scholars had puzzled over this artistic choice. Why would an ancient Egyptian artist show their pharaoh in such a vulnerable position? Now they had the answer. The artists were simply depicting reality. The DNA evidence confirmed what ancient art had been trying to tell us all along. The genetic analysis also provided compelling evidence about Tutankhamun's cause of death. For thousands of years, his death remained shrouded in mystery and speculation. Some proposed murder, others suggested a chariot accident, but the DNA revealed markers indicating he suffered from repeated malaria infections. In our modern world, malaria is treatable with medications, but 3,000 years ago, there was no cure. Repeated bouts of malaria, combined with his compromised immune system due to genetic disorders from inbreeding, created a deadly combination. The young pharaoh's body simply couldn't withstand the constant assault of the disease. At just 18 years old, his reign ended and Egypt's history changed forever. He had ruled for only 9 years, ascending to the throne when he was merely 9 years old. These were the genuine findings from the 2010 JAMA study, and they were fascinating in their own right. They revealed a complex picture of royal life in ancient Egypt, the consequences of dynastic power struggles, and the biological realities of ancient pharaohs. This was legitimate, peer-reviewed science that added significantly to our understanding of history, but somewhere along the way, something went wrong. The story took a turn that the original researchers could never have anticipated. The truth got lost in a fog of sensationalism, misunderstanding, and deliberate misrepresentation. The viral claim emerged that Tutankhamun's DNA didn't match any living human on Earth. The internet exploded with theories. Some claimed he was an alien. Others insisted he was European royalty. Conspiracy videos amassed millions of views. The claims became increasingly outlandish, and the original scientific findings disappeared beneath layers of speculation and hype. But where did this sensational claim actually come from? The answer leads us to a Swiss company called iGena, a commercial DNA testing firm. The company saw an opportunity in the publicity surrounding the 2010 JAMA study. They decided to launch what they called the Tutankhamun DNA project. Here's where the story becomes particularly problematic. The company didn't have access to the original DNA samples or the detailed scientific data. Instead, they based their analysis on something far more questionable. They watched the Discovery Channel documentary about Tutankhamun's genetics. They took screenshots from the television broadcast. From those screenshots, they attempted to reconstruct Tutankhamun's Y chromosome profile. This methodology is so scientifically questionable that it's almost difficult to believe it happened. Imagine trying to conduct serious genetic analysis by photographing images on a television screen. Yet that's essentially what iGena did. From these television screenshots, they claim to have identified 16 genetic markers on Tutankhamun's Y chromosome. Based on this reconstructed profile, iGena announced that Tutankhamun belonged to haplogroup R1b, specifically the subclade R1b1a2. They claimed this meant he was related to over half of all Western European men today. The headlines were sensational. Media outlets around the world picked up the story. Tutankhamun, the ancient Egyptian pharaoh, supposedly had European DNA. The conspiracy theories began almost immediately. If he was genetically European, how did he end up ruling Egypt? Was this evidence of a lost European civilization? Was it proof of extraterrestrial intervention? Did it prove some kind of racial theory? The claims spiraled further and further from reality. Videos went viral on YouTube with millions of views. Reddit threads exploded with speculation. The claim that his DNA didn't match any living human became widespread internet folklore. But here's the critical problem with all of this. The peer-reviewed scientific community immediately pushed back. Dr. Albert Zink, one of the actual co-authors of the original 2010 JAMA study, examined Igenea's claims directly. He reviewed the television footage that the company had used as their source material. His conclusion was damning. Zink stated that Igenea was acting in a manner that was fundamentally unscientific. The company had overstepped the bounds of what their data could support. They had made claims far beyond what any responsible scientist could make from television screenshots. Multiple population geneticists criticized the Igenea interpretation as purely speculative and methodologically unsound. The problem runs deeper than just methodology. The original 2010 JAMA study, while groundbreaking in many ways, did not actually publish the detailed Y chromosome data needed to make precise haplogroup assignments. The researchers didn't release SNP-level detail, the specific genetic markers necessary to confidently determine which haplogroup Tutankhamun belonged to. Without this crucial data in the published record, no one could definitively verify what haplogroup he carried. Think about what this means. A commercial DNA company made a precise genetic claim about an ancient pharaoh based on images they photographed from a television documentary. They made this claim without access to the original research data, without the detailed genetic markers, and without following standard scientific protocols. And millions of people around the world believed it. The claim that Tutankhamun's DNA didn't match any living human became accepted as fact in popular culture, despite having no legitimate scientific foundation. This is a fascinating case study in how science gets distorted, how commercial interests can exploit public curiosity, and how misinformation spreads in the digital age. When you search for Tutankhamun's DNA online today, you'll find countless websites, videos, and articles repeating the Igenea claims as if they were established fact. Many of these sources don't even mention the controversy or the scientific criticism. They simply repeat the sensational claim as though it's proven truth. Now, if the R1b1a2 haplogroup claim hasn't been verified, what should we actually make of it? Even if Tutankhamun did carry haplogroup R1b, what would that really mean? This is where we need to understand what haplogroups actually are. A haplogroup is a group of haplotypes that all descend from a common ancestral haplotype. On the Y chromosome, which is passed from father to son, a haplogroup traces patrilineal ancestry. It represents a single paternal line among thousands of ancestral lines in any individual's genetic makeup. It is emphatically not an ethnic label. It is not a statement about someone's overall ancestry, their cultural identity, or their genetic makeup as a whole. Haplogroup R1b is indeed common in Western Europe today, found in roughly 70% of men in some parts of Western Europe. But this tells us only about one specific paternal lineage. It says nothing about Tutankhamun's maternal ancestry, his autosomal DNA, or his overall genetic composition. Moreover, the geographic distribution of haplogroups is complex and cannot be simplified into neat categories like European or African. Ancient Egypt sat at a geographic crossroads linking northeastern Africa and Western Asia. Archaeological evidence demonstrates long-term interaction between Egypt and the Levant, Nubia, and the Eastern Mediterranean. Populations moved, traded, and intermarried across these regions for millennia. A Y chromosome haplogroup carried by an 18th dynasty Egyptian pharaoh would need to be interpreted within this complex historical and geographic context, not as some kind of smoking gun proving European ancestry or a hidden connection to modern Europeans. Beyond the haplogroup controversy, the actual DNA findings from the 2010 study tell a comprehensive story about Tutankhamun and his family. The genetic analysis confirmed that Tutankhamun's parents were full siblings, meaning they shared both a mother and a father. This wasn't particularly surprising to Egyptologists, as historical records suggested that royal incest may have occurred multiple times within the royal family. But having genetic confirmation was significant. The study further traced the family tree back through 5 generations, identifying grandparents and great-grandparents. Through genetic testing of multiple royal mummies, the researchers determined that Tutankhamun's great-grandparents were the pharaoh Amenhotep III and his queen Tiye. His grandparents on his father's side were Yuya and Thuya, whose mummies were discovered in tomb KV46 in the Valley of the Kings. This comprehensive family tree, established for the first time through DNA analysis, provided unprecedented insight into the royal family's structure during the 18th dynasty. The consequences of generations of interbreeding within the royal family became increasingly evident in Tutankhamun's generation. Beyond his deformed left foot, he carried genetic markers indicating increased susceptibility to disease. The repeated malaria infections that ultimately killed him represented an interaction between the pathogen and his genetically compromised immune system. Interestingly, Tutankhamun was not particularly unusual in this regard for the royal family. His parents likely experienced similar health challenges due to their shared genetics. This raises fascinating questions about the long-term consequences of dynastic succession practices. If royal families consistently intermarried to maintain bloodlines, did this gradually weaken the genetic fitness of successive generations? Did the health consequences of inbreeding eventually contribute to the decline of dynasties? These are complex evolutionary and historical questions that the DNA evidence can help us explore. The scientific study also examined 2 mysterious female fetuses found in Tutankhamun's tomb. Both were approximately 5 to 7 months in gestational development at the time of death. DNA testing revealed they were Tutankhamun's biological daughters, offspring from his marriage to his wife Ankhesenamun. The fact that both fetuses appeared to be female and both failed to develop to term suggests possible genetic incompatibility or developmental issues. Neither child was born alive. This added another tragic dimension to Tutankhamun's brief life. Not only did he suffer from genetic disorders and disease, but he and his wife were unable to produce a surviving heir. His death without issue meant the end of his particular royal line and contributed to the succession crisis that followed his reign. These findings humanized Tutankhamun, revealing him not as a distant historical figure, but as a young man struggling with physical ailments, genetic disorders, and personal tragedy. The 2010 study also produced a family tree that revealed surprising information about several disputed identities. For centuries, Egyptologists had debated which mummy belonged to which pharaoh. Some identities remained controversial because pharaohs were sometimes buried in tombs other than their own, and tomb labels could be misleading or lost to time. The DNA evidence helped clarify some of these mysteries. It confirmed relationships that historians had long suspected and challenged certain assumptions about royal family membership. However, the study also raised new questions that remain unanswered. The identity of the KV-55 mummy, assumed to be Akhenaten, cannot be confirmed with absolute certainty through DNA alone. The DNA showed he was Tutankhamun's biological father, but Egyptologists still rely on historical records and other evidence to identify him as Akhenaten specifically. This demonstrates that while DNA analysis is incredibly powerful, it works best when combined with archaeological, historical, and other scientific evidence. Why did the Igenia claim spread so widely despite lacking scientific support? Several factors contributed to the viral success of the conspiracy theory. First, the original findings were genuinely fascinating, which meant media outlets covered the story extensively. This extensive coverage created public awareness and curiosity about Tutankhamun's DNA. Second, the findings challenged conventional understanding in interesting ways. Incest in the royal family, genetic disorders, and disease all seemed shocking and dramatic. This novelty made for good storytelling. Third, the commercial claim that his DNA was uniquely European or didn't match living humans was sensational enough to grab headlines even more aggressively than the original peer-reviewed findings. News organizations, particularly those focused on mysteries and unexplained phenomena, found the commercial claim irresistible. Fourth, the internet ecosystem amplifies sensational content. Videos making outlandish claims about Tutankhamun's extraterrestrial origins or European heritage accumulated millions of views. YouTube's algorithm rewarded engaging content regardless of accuracy. Conspiracy theory communities embraced the narrative enthusiastically. Additionally, there's something psychologically compelling about conspiracy theories. They suggest that official science is hiding the truth, that mainstream media is lying to us, and that the real story is far stranger than we're being told. For some people, this framing is more appealing than the actual scientific findings, even though the actual science is genuinely fascinating. The idea that we've been deceived creates a sense of special knowledge and community among believers. The conspiracy narrative also intersected with broader ideological debates about race and European cultural identity. Some people use the R1b haplogroup claim to support arguments about European superiority or to challenge narratives about African civilization. These ideological motivations ensured that the claim would persist even in the face of scientific criticism. It served a purpose beyond its factual accuracy, which meant people had reasons to keep believing and spreading it. It's worth noting that the original 2010 JAMA study itself faced some criticism from the scientific community, though for different reasons than the Igenia claims. Some researchers felt the authors didn't release enough raw data for independent verification. Others questioned certain methodological choices. This is normal in science. Peer review is an imperfect process, and legitimate disagreements about methodology and interpretation happen regularly. However, the core findings about family relationships, health conditions, and the cause of death have held up well to scrutiny. Subsequent E. Research and re-examination of the data have generally supported the original conclusions. This is how science is supposed to work. Claims are made, they're subjected to critical examination, and they're either supported or challenged by further research. The iGenia claims, by contrast, have not held up to scrutiny. When geneticists have examined them critically, they've found them lacking in rigor and unsupported by the available data. The Tutankhamun DNA story also highlights the importance of scientific literacy in our digital age. As ordinary people, we're constantly bombarded with claims about scientific discoveries. Some come from legitimate sources, and some don't. How do we distinguish between the two? One key indicator is whether claims are published in peer-reviewed journals. The 2010 JAMA study went through rigorous peer review before publication. Experts in the field examined the methodology, evaluated the data, and assessed the conclusions. Only after passing this scrutiny was it published. The iGenia claims never went through this process. They were announced directly to the media without peer review. Another indicator is whether scientists working in the field support the claims. Dr. Zink's criticism of iGenia's methodology was significant precisely because he was one of the actual researchers who conducted the original study. When the scientists themselves say something doesn't add up, that's a red flag. Additionally, we should be skeptical of claims made by commercial companies with financial incentives to attract attention. iGenia was promoting a commercial DNA testing service. Sensational headlines help sell services. Peer-reviewed scientists have different incentives. Their reputation depends on the accuracy and rigor of their work. The media's role in spreading the Tutankhamun DNA myth is also worth examining. Many news outlets reported the iGenia claims without adequately conveying the scientific criticism. Headlines screamed about Tutankhamun's European DNA without mentioning that geneticists disputed the findings. This isn't necessarily malicious. Many journalists lack the scientific expertise to evaluate complex genetic claims. They rely on quotes from experts and established sources. But when a commercial company makes a sensational claim and legitimate experts criticize it, responsible journalism should convey both sides of that controversy. The public deserves to know not just what was claimed, but whether those claims hold up to scientific scrutiny. Some media outlets did provide this balanced coverage, but many didn't. The sensational version spread much farther than the nuanced version, which is how myths are born. What we're left with is a fascinating case study in how science gets communicated, misunderstood, and distorted in the modern world. The actual scientific findings about Tutankhamun's DNA are genuinely remarkable. They tell us about family relationships that were hidden for thousands of years. They reveal the genetic consequences of royal inbreeding and the health challenges that even the most powerful people in the ancient world couldn't escape. They provide evidence about the cause of death for a historical figure who has captivated human imagination for over a century. They confirm what ancient art was trying to tell us about his physical condition. These are profound scientific achievements that deepen our understanding of history. Yet the narrative that dominates popular culture is the false claim about his DNA not matching any living human. The truth got buried beneath layers of sensationalism. As we continue to develop more sophisticated tools for understanding ancient DNA, we'll likely discover more secrets about historical figures and ancient populations. These discoveries will continue to reshape our understanding of history, but they'll only serve that purpose if we approach them with appropriate skepticism and scientific literacy. We should celebrate genuine scientific breakthroughs while remaining critical of claims that seem too sensational to be true. We should seek out peer-reviewed research and understand that published science is not the same as news stories about science. We should recognize that commercial interests may distort scientific information, and that experts in the field are usually the best sources for understanding complex scientific claims. Most importantly, we should remember that the actual truth is often more fascinating than the myths we create about it. Tutankhamun's real DNA story, with its revelations about royal incest, genetic disease, and a young pharaoh struggling against biological odds, is far more interesting than any alien conspiracy theory. The challenge is ensuring that these real stories reach people's ears louder than the sensationalized myths.